All right, well, greetings, everyone. I think we should go ahead and get started. People are still logging on, but we want to make sure to have adequate time for our two speakers. So very pleased to have Matt Weber from Notre Dame and Stephanie Stanford from UC San Diego with us today. Matt's going to lead off. He has had an Accelerator Award since 2019, and he's going to tell us about improving glucose control with applied supramolecular chemistry. Matt, please.
Great, thank you for the introduction. I'll go ahead and get fired up here. All right, so I'd like to thank the Pathway group, especially for organizing these. I've been participating in them, with these monthly calls, and I've found them to be very useful just in terms of expanding my own knowledge of the broad and evolving space of diabetes. And so I'm happy to be here and present my little slice of this particular research area. I'll note that this talk comes at a actually fantastic time for me. The first reason, and I'll tell you the second one later, the first reason is that it's the first talk I've given as a tenured associate professor. And this is something effective as of July 1. And I think the Pathway Award was a very key piece to this actual career transition. And so I'm excited to be able to give kind of my first talk following tenure to the Pathway group. And so I think that's been very important. So I'd like to start where I intend to end, and that is in thanking, my research group, primarily the people who actually do the work. I just get to be here and crack jokes and, talk about their work, but of course they're the ones who do the work. And so I'd like to thank my research group. And I've highlighted here with a red arrow, my graduate student, Sihan Yu, who has been really leading a lot of the efforts in some of our evolving interests in glucagon delivery that I'll tell you the story about today. And so I'm excited about that. And then of course, thankful as well for funding, primarily the Pathway Award, which I'm grateful for, especially, amongst this audience, but then, funding for a lot of our other works in other ways of treating diabetes with insulin or with dual hormone therapies or other general drug delivery strategies or new materials design strategies or different things like that. And so I'm grateful for all of these different forms of support that support our research in various capacities. So I think it is possible that, to explain to this audience and shouldn't require too much convincing that we have a remaining glucose control problem in treating diabetes. And so I took this data here from the supplement of a paper in the New England Journal of Medicine a handful of years ago. And this was a paper that was evaluating the best, at the time, control algorithms for a dual hormone pump to manage diabetes, one of these learning algorithm type situations. And what you'll see from this few days of blood glucose traces following sort of the learning algorithm, right, is you still have some highs that are approaching kind of the unacceptably high range, and you have some lows that are getting into kind of the scary low range. And so even under what we would consider kind of an optimal control situation by other members of my field, this is actually work done by chemical engineers, right? We still have these issues with blood glucose control. And of course, this leads to a variety of both acute and long-term complications, challenges, ranging from, the types of things that'll affect you, 15, 20, 40 years down the road, to the things that might affect you tonight, right? The very scary sort of acute complications. And so there remains a need for better strategies to control blood glucose and diabetes. And so in my field, the kind of field of drug delivery and materials design, there's been a variety of efforts that have sought to come up with new technologies to address this blood glucose control challenge. Of course, there's evidence in the fields of cell replacement using transplantation or stem cell sourced glucose sensing insulin secreting cell populations, to be able to encapsulate these and facilitate glucose sensing using a biological approach. The field we've kind of termed the bionic pancreas, the idea that you could have a series of hardware, sensors, pumps, and then some sort of control algorithm run on your standard iPhone or whatever it might be, that would actually facilitate blood glucose control using kind of state-of-the-art mechanical and sensor-based technologies. And then kind of the area that we've been working in since my postdoctoral training and then extending into my independent career is the idea of smart drug delivery, which we've kind of termed the fully synthetic pancreas, that we could make insulin or materials that could themselves sense blood glucose and modulate the potency or modulate the bioavailability of insulin as a function of need being the blood glucose level. And so I use a word on the title slide that maybe is not familiar to a variety to various people in the audience today. That is the phrase supramolecular chemistry. And so this is the field of chemistry that I received my foundational training in my PhD work. This is the idea of doing chemistry beyond the molecule, leveraging specific non-covalent and dynamic molecular recognition motifs in order to facilitate organization of molecules without the need for covalent bonds. And so this is kind of the examples of, at least in my lifetime, some Nobel laureates that have received prizes directly or somewhat indirectly related to this concept of supramolecular chemistry. So where do I see that we could use supramolecular design for better therapeutics, specifically as it relates to diabetes? And so this has been the area where we've been really kind of thinking about applying some of this non-covalent sensing, equilibrium governed chemistry to the space of diabetes therapeutics. And so there could be a bunch of benefits to this approach, right? If I have non-covalent associations of molecules, these are highly dynamic. They're governed by thermodynamic principles. We could have things that could exchange rapidly, reversibly, and with predictable sort of kinetics on and off rates, these kinds of things. We could think about making motifs, molecular scale motifs that are tunable, and enabling this to tune the chemical, biological, if we make these into materials, the mechanical properties of the materials, et cetera. I like the idea also of thinking about these in the context of modular platforms, specifically the idea that we could make person-specific approaches to personalized medicine, right? I could take a supramolecular motif, keep that conserved, and then engineer away from that motif to include specific features or ratiometric presentation of signals that might be specifically tuned for a person. If I don't have to overcome this energy of a covalent bond, I could do more rapid sensing of environmental cues. Of course, the cue we care a lot about in this work would be glucose. And then thinking about making materials or protein formulations or things that could exhibit aspects of biomimicry or even dynamic reciprocity that could talk to a living dynamic world and then respond and adjust their properties in kind. And so I got my start in this area thinking about aryl boronate chemistry or phenylboronic acid chemistry, which is able to use a chemical motif of phenylboronic acid that can bind reversibly to cis-1,2 or cis-1,3 diols, such as glucose. The binding of these is highly variable. It's not extraordinarily specific for glucose, which leads us to a challenge that we're trying to address in some of my other research projects ongoing that I won't speak about today. But generally speaking, this is a way to use the binding of glucose to stabilize the negative charge and facilitate some sort of change in relative hydrophobicity of a ligand, for instance, or use glucose diol cross-linking in order to stabilize the material. And so we started with this by engineering insulin with a glucose binding motif attached to the B29 lysine residue, putting one of these groups on and making a molecule that looks a lot like insulin detamere, except has this phenylboronic acid on here. We'd evaluated this in a mouse model, STZ diabetic mouse model, injecting these at time 0 here, various modified insulins as well as native insulin, and then simulating throughout the course of a day something looking like meals, right? An intraperitoneal glucose tolerance test, a series of these 3 times over the course of a day. And we can get out here, we're 10 hours following the administration of these insulins. We simulate one of these glucose challenges, and we have insulin candidates that actually continue to reverse blood glucose back to a normal glycemic range. And so in doing a lot of this insulin modification chemistry, we got sort of interested in ways that we could modify insulin and modulate its properties non-covalently. We thought this could be a more scalable approach, more of a formulation-centered approach. So that got us into this branch of chemistry using macrocyclic recognition of N-terminal aromatic residues, and specifically the B1 phenylalanine residue on insulin, which is actually a very good guess for this macrocycle that my group has grown a bit affectionate for, this curcubit 7 ureal. So this binds with OK binding affinity on the order of about 10 to the 6th inverse molar, which is just good enough to work in a vial for formulation purposes and just bad enough to not really work in the dilute environments of the body. And so this was a little bit of a both an opportunity and a challenge for what we could do with this, or sort of a limitation of what we could do with this that we've continued to explore. And so working with my good friend and colleague, Eric Apple, who's now faculty at Stanford, both of us were postdocs together at MIT at the time. And Eric's continued on to do great stuff in diabetes related work for protein formulation as well, both in collaboration with my group and then on his own. We were looking at the N-terminal modification non-covalently of insulin with a pendant polymer to facilitate its stability and formulation. So the first thing we did was we put a polyethylene glycol on here. It's a great polymer for preventing protein aggregation or opsonization or things like that in the body. But it also stabilizes things in formulation. And so we could take insulin formulations, stabilize these out to 100 days in stressed aging conditions. And even after 100 days, we could take this insulin and it would maintain its activity similar, basically identical to the freshly dissolved insulin we had just made. We've demonstrated, working with Eric's group as well, the ability to make rapid acting monomer formulations of insulin. So by pulling out the zinc and actually monomerizing the insulin that would otherwise be very unstable can be very stable and maintain monomer form for more rapid action upon administration. And then also we've demonstrated, even in diabetic pigs, the ability to use this additive as a formulation excipient to stabilize co-formulations of insulin and pramlintide, which is the amylin analog, which otherwise you can't really get into the same vial because of pH incompatibility. But when we add this in, we're actually able to get insulin and pramlintide in the same vial, leading to a more biomimetic pharmacokinetic profile of the two proteins, which in a normal beta cell would be secreted at the same time. In type 1 diabetes, of course, the amylin signal is lost and not frequently replaced by therapeutic regimens. And so we think this is one example of using supramolecular chemistry as a strategy to stabilize insulin, enable co-formulation with synergistic hormones. My group is presently looking at this strategy for better safety profiles in glucose responsive insulin hydrogels and other sorts of things as well. And so this is some interesting chemistry that we've really been exploring quite a bit. Suffice to say, I'd like to kind of keep the insulin work short for this particular talk, the duration of this talk, and really kind of focus on where the remaining challenges in drug delivery related to diabetes are found. And so I think that there's a need to improve the availability and release of insulin as a function of blood glucose. I think a lot of the technologies that have been developed have either a delay that is kind of unacceptable, or they leak. Right, so you get blood glucose restored by releasing insulin, and now your insulin keeps coming out. You get, hypoglycemic periods following a period of need. I think we need to think about better strategies to ensure compatibility of materials or modifications that are used. I'm going to tell a story that contradicts this point a little bit, but a lot of times we're relying on exogenous proteins, exogenous enzymes, materials that lack acceptable biocompatibility or safety profiles. And so I think we really need to think about this moving forward as we continue to think about clinical translation of drug delivery strategies in the diabetes space. And then, advancing towards more bio-inspired insulin therapeutics. So one example would be, like I showed, co-delivery with synergistic hormones, potentially liver-directed functions or of glucose-responsive insulins. So getting more of a mimetic profile of exposure in tissue through formulation or protein engineering. And then thinking about integrating better this abiologic-biologic communication and sensing platforms. And so I would say that perfect glycemic management with synthetic approaches is still very, very complicated, still depends on a lot of factors. And this is really just the challenge for insulin. And there's 30 plus years of people like me in the drug delivery space working on better ways to deliver insulin. And so this kind of leads to where I'm going to pivot and talk more about glucagon at this point. And glucagon delivery remains almost completely unexplored. And so the first question I wanted to ask, as we kind of thought about how we would deliver glucagon, what would it even look like? How would we think about glucagon? Could we think about this as a synthetic corollary to the dual hormone pump? So I could take my past sort of phrasing of the fully synthetic pancreas and plug a glucagon element into one synthetic module and an insulin element into the other synthetic module, and these could sort of have kind of the yin and yang release profiles as needed. So that could be one possible option. The other option is we could think about a glucagon drug delivery concept in the context of a prophylactic device. or I could have a gel or a material or a microneedle patch or something like that on board with glucagon at the ready, just in case you have some hypoglycemic episode. And so I think that these are sort of the two possibilities. And there might be kind of an area in the middle of these even still, where you could have glucagon around sort of as a basal support for better control, but then it's there also to prevent severe hypoglycemic episodes as well. And so I think that there's some different ways we could think about this. And so where we kind of started thinking about this is going back to our roots in understanding supramolecular chemistry and really the concept of engineering the supramolecular landscape. And so this comes from a position, from sort of a perspective of bioinspiration, where we can look, at very complicated free energy profiles of protein folding, for instance, and we can see, a lot of downhill slopes, but we can see some valleys, some different peaks, some local minima as well as global minima. We can start to understand that we could design materials the same way. We could have materials that exist in sort of a global free energy minimum. We could have materials that exist in states of kinetic arrest of some sort, or we could have states that are somewhat metastable and just, we look at them a little bit longer and all of a sudden they've changed on us, right? And then there's a fourth option, which is we could actually engineer materials that exist in states out of equilibrium. And so we could use the consumption of some fuel to drive materials into a non-equilibrium state, wherein they are stable only if that fuel is around, and if the fuel is gone, the material would fall apart. And so we wanted to ask the question of what if such a fuel was glucose? And so our thinking would be, we could take glucose and use this as this fuel to push this ball up a hill, right, where the ball is inherently unstable, and will fall back down if glucose is absent, causing the material to fall apart. And so this, like I said, comes from a place of bioinspiration as well. We could look at examples of chemically fueled non-covalent polymerizations in nature. Things like the actin cytoskeletal elements that polymerize and depolymerize as a function of availability of ATP, or microtubules that do basically the same thing as a function of the availability of GTP, right? And so we can get remarkable function, the function that underlies the mobility of all of our cells, the mechanical properties of our cells, from this type of chemically fueled non-equilibrium assembly of molecules. So this led us to our first efforts in this, and why this is also an exciting time for me is this paper was actually just accepted this morning. And so I'm very happy to be able to talk about it. We got the patent, provisional patent filed yesterday, paper accepted today. And so it's been a good week to be able to present this story, which is why I'm excited to tell it. Thinking about this idea of using materials that assemble in the presence of glucose, in this case, actuated by an enzymatic sensor, so glucose oxidase, that would drive glucose to gluconic acid, use the concomitant drop in pH to transiently stabilize materials. But if my glucose is gone, if this glucose fuel is gone, my materials fall apart and dissipate back into sort of a dissolved, unstable state. And so the thinking with this is we could actually use these materials to encapsulate glucagon, use the presence of resident glucose in tissue to stabilize the material. And if the blood glucose drops below a certain threshold level, the material now doesn't have adequate fuel to stay assembled, it falls apart. And so our first task was really to kind of identify a material that could even do this as a function of pH before we even thought about integrating glucose. And so we developed a molecule that could self-assemble and form self-supporting gels. These are very easily injectable gels, but they are stable at low pH. So the kinds of pHs, pH 5, for instance, that you might see in a local microenvironment from glucose oxidase. But at pH 6, 7, 8, they start to flow, right? And we can measure this through what we call kind of poor man's rheology. We could have the vials upside down or not and determine if they're gels or not. Or we could do things where we actually quantify the mechanical properties and the network connectivity here, where we see a dramatic drop-off, orders of magnitude drop-off in the network connectivity of these materials as a function of pH, right? Where we go from pH 5, very, very stiff, very stable, highly networked materials, 2 orders of magnitude drop as soon as we go to pH 6, and another order when we go to pH 7. We then took these materials and we converted that stimulus from pH to glucose. And so we could make materials that flowed in the presence of 0 or 50 milligrams per deciliter glucose, but were pretty stable gels in the presence of 100 or more milligrams per deciliter glucose. And you can see this example again, looking at the network connectivity, the storage modulus of these materials over time. So the next thing we wanted to do was actually take a glucagon and encapsulate it within this material and demonstrate, or at least see if we could demonstrate glucose responsive release from a material. And so we take the materials and we make them in the presence of glucose at the low pH environment that we think they would be in. And then we incubate them in different amounts of sort of bulk buffers containing different amounts of glucose. And so if we have these gels and we put them into buffers containing no glucose, we get more release than if the buffer contained 50 milligrams per deciliter than if it contained 100 or 150 or 200, for instance. So we can actually get release of, in this case, dasiglucagon, which we use as a stable analog of glucagon, as a function of blood glucose, where we get more release in the presence of lower amounts of glucose, which is sort of the reverse of what everybody has done so far in this field in the context of insulin release. We're able to kind of reverse engineer this system to actually release glucose in the present, release glucagon in the presence of low glucose. We can come down here and we can simulate this idea of sort of a sudden hypoglycemic event where we go, from 100 milligram per deciliter where we have kind of low level release to 0 milligram per deciliter where my release now accelerates pretty rapidly. And so we're kind of excited about this. We realize it's not perfect. It's not perfect for a few reasons, right? We get some leakage at times when you maybe wouldn't want it, 200 milligrams per deciliter. We get a little too much release there. And the release we do see is a little bit too slow. Right, we'd like to see release, especially if we think about in a prophylactic capacity, we'd like to see, sudden release on the order of, half an hour, something like that. And we're not quite there yet, but we do feel pretty good about the idea that we could actually modulate release as a function of blood glucose from this system. So the thing we then went and tried was actually a mouse model, and we had to really kind of develop our own mouse model for this space. There wasn't a ton available to us to really go to the literature and look at what we might want to do. And so we start with, STZ mice that are highly diabetic, blood glucose levels in the range of about 550 milligrams per deciliter. We first give them detamir to get them onto a plane of normoglycemia using a basal insulin analog. And so the nice thing about detamir is it can get them down to, sort of about what a normal mouse would be, 180 or so milligrams per deciliter is normal for a mouse. We then treat them with our gels containing dasiglucagon alone, or buffer in this case, and we see some leakage from the gels. And so this is another point where our materials are not yet ideal, but we see some leakage from the gels, whereas we get some rise in blood glucose corresponding to dasiglucagon activity. But then a couple hours after we've given these gels, we challenge these mice with an insulin overdose, IP, so it hits pretty fast. And we measure hypoglycemia over that time following that insulin overdose and look at the difference between hypoglycemia of our untreated versus our treated mice, including the evidence of some deaths even in these mice that are treated with dasiglucagon or buffer. Whereas our gel with dasiglucagon induces no death, the mice on average stay above a threshold value that we sort of artificially set of 60 milligrams per deciliter. We can go over here, we can look at nadir blood glucose levels and see significantly higher blood glucose levels compared to these controls. And our final blood glucose levels are also higher compared to controls. And so we're pretty excited about this as a first sort of platform to understand how we might use glucose to engineer stability in materials that then fall apart in the absence of glucose. Thinking about this maybe moving forward, where are we going next? We have some ideas for things that are exploding nanoparticles that actually could explode and release all of their payload in very short times, on the order of maybe an hour, for instance. This is one platform we're evaluating. And then even can we do this without enzymes? Could we engineer molecular self-assembly to proceed without the need for an enzyme to actuate that signal, just by binding glucose in specific ways? And so we've actually demonstrated kind of as a first pass the ability to use glucose as a gelation agent of these types of materials, where if we don't have enough glucose, they flow. If we do have enough glucose, they don't. And using this as a way to stabilize these materials without the need for the enzyme. And so we think maybe we could make these into injectable hydrogels as we demonstrated. We've also begun some work to fabricate microneedle patches or arrays of these so we could actually have them be easily wearable on the skin. And so this is some things we're looking forward to kind of reporting over the next couple of years. And so with that, I'll end where I started. Thank my group, specifically Hannah, and the funding sources for support of this work. Thank you.
Wow, that was really exciting. Wonderful. So let's go to a question from Rich Bergman. Is it possible to regulate blood glucose with glucagon alone, or will it work more as an emergency measure? How are you looking at that?
You know, one of the funny things about glucagon that we actually learned a little bit is if we did these studies in healthy mice, it actually stimulates insulin secretion. And so we could actually get more if you go at very high doses. And so this was something we actually learned was in healthy mice, we stimulate them with dasiglucagon, we actually got increased insulin secretion and increased hypoglycemia. I don't know that it's possible alone, but I do know that there are people right now. There's a company that started recently that is actually looking at co-formulating insulin and glucagon in the same vial. And what they're thinking is that they can actually prevent the hypo by having the glucagon around all the time, while not compromising the ability of insulin to control the hyperglycemic events. So probably you still need insulin, especially in type 1, but I think there could be possibilities of using glucagon in more sort of intelligent approaches for glycemic control.
So maybe you said this, but I'll ask you to clarify at a minimum. So you have a beautiful glucose regulated capsule assembly that you've devised. And of course, the way it is set up now is ideal for glucagon. Glucagon appears in the system at low glucose and it's trapped in your devices at high glucose. Is there a way to engineer that in reverse to facilitate insulin release?
Yeah, no, I think that's pretty straightforward. I think we could do that. I think it would just require altering some of the charge states of different molecules in our gelators, our small molecules. It's something we've talked a little bit about doing. We kind of went after the glucagon challenge first because we thought it was harder and we thought it was maybe a little bit higher impact and more interesting. But there are people that have used glucose oxidase to similarly gel or other materials in the reverse way that we're doing it. Yeah.
Do you think there's any limitation or maybe advantage to implanting these glucagon devices in certain regions in the body? Have you thought about sub-Q versus? I'm not sure. Other places?
You know, all we've looked at so far is sub-Q, just kind of both because of ease of access. I mean, also we're engineers and we're not so good at, injecting mice with things. And so sub-Q is reasonably easy for us. But also just thinking about if you put this in the hands of a person with diabetes, right? The sub-Q injection or even intramuscular injections, these kinds of things are pretty routine. And so I would be a little, especially if we thought about this as like a daily or every couple of daily injection, I think you'd want to have it be accessible for at-home application.
Great. And a question from Dr. DeLong: how will this immune system respond to such devices? Do you have any sense of that yet?
I think, unfortunately, probably not great at this point. And I say this specifically relevant to the glucose oxidase switch in here, or the sensor in there. I don't know that that's going to be translationally feasible. And so that's, being, a full candor, I think that we need to engineer that piece out of it. And so that's why I'm excited about some of these later manifestations that I think we could engineer that piece out to improve the immune tolerability of the glucose oxidase. I think if we had used glucose oxidase, we'd want to encapsulate it in some polymer capsule or something like that to try to keep it from being fully visible to the body. But I do worry about these xenogenetic sourced enzymes and any sort of immune compatibility of these things.
That was a good question. All right then. Any last comments, questions? If not, I think we'll move on to our second speaker. Thank you so much. That was truly,
okay. No, thank you.
That was really creative, and congratulations on your paper. That's great news.
Yeah, it's a good day. So thank you.
No doubt. Okay, so our second speaker is Stephanie Stanford from UCSD. And Stephanie was an initiator category awardee in 2015. So Stephanie, you're almost done with your award period, so we expect a lot from you. Come on now.
Oh boy. Well, first of all, I thank you. And I'd really like to thank Chris and Kylie for organizing these monthly meetings. I think this is a great opportunity, especially for us to meet and get to hear about each other's research, especially during this pandemic time. And Matt, that was a great talk, very applied, very interesting. My talk is very much more basic biology, as you'll see, about a phosphatase that promotes diabetes, trying to understand what this phosphatase does in adipocyte biology. And so it's about the LMPTP, which is a tyrosine phosphatase that we found promotes adipogenesis and subcutaneous adipocyte hypertrophy. And so LMPTP is basically a metabolic syndrome gene. It's encoded by the ACP1 gene, and there are genotypes in humans which encode for different amounts of total LMPTP enzymatic activity. And people who have lower LMPTP enzymatic activity have protection against hyperglycemia and dyslipidemia in obesity and also during diabetes. And this gene associates with clinical variability in obesity. And we had previously done studies on LMPTP and high-fat diet diabetes model, and we found that LMPTP acts as a promoter of insulin resistance and diabetes symptoms in high-fat diet-fed mice, and that's through an action on the liver. We had conditional knockout mice. We found that when we deleted LMPTP only in the liver, it protected them from insulin resistance and gave them normal glucose tolerance in high-fat diet. Now, so what we found is that LMPTP acts as an inhibitor of insulin signaling in the liver during obesity. And if you treat high-fat diet-fed mice with an LMPTP inhibitor that we had generated, it reverses insulin resistance and it improves glucose tolerance. So it recapitulates the effect of the liver-specific LMPTP knockout. Now, in that study, we found that if we knocked out LMPTP in the adipose tissue, we didn't get an improvement in glucose tolerance, but we had an interesting phenotype that we wanted to explore after. LMPTP is highly expressed in adipose tissue. It's a ubiquitous phosphatase, but it's very highly expressed in adipose tissue. And there's just was not much known about the biology of LMPTP in adipocytes. And what we found is that if you specifically knock out LMPTP in the adipose tissue using adiponectin Cre, we found that the adipocytes were smaller. So you can see the Cre negative mice on high-fat diet, they get these this is subcutaneous adipose tissue. You can see the very nice big adipocytes. We found that there seemed to be more numerous but smaller adipocytes when we knocked out LMPTP that you can see in the Cre positive mice. Now we found this in the subcutaneous tissue, but we did not see this in the epididymal fat tissue. And so you can see here these graphs, the adipocyte size is smaller, and you can see from this frequency distribution that we had skewing in the LMPTP conditional knockout mice towards the smaller adipocytes. So we then started to look into what's LMPTP doing in the adipocytes. So we looked to see if LMPTP was affecting the generation of these cells from fibroblasts. So first what we did was we start over here on the left, you can see we took fibroblasts from the fat pads of LMPTP knockout mice, and we differentiated them in vitro using an adipogenic cocktail. And what we found is that while the wild type cells differentiate into adipocytes, the knockout cells were deficient in this process. So then we knocked down LMPTP using a cell permeable antisense nucleotide that actually just goes right into the cells in the cultured cells. And we did this both in 3T301 preadipocytes and also primary human preadipocytes, and we from visceral fat tissue. And we found that in both cases, if you knock down LMPTP, there's again impaired adipogenesis. And then to understand if this was due to LMPTP's phosphatase activity, we used an inhibitor of LMPTP that we developed. Actually, we used two different ones. Our original kind of hit compound that we'd worked on, which we call compound 3, and then a later more potent derivative, compound 23, which is the one that we used in our diabetes study in mice. And using both of these, we found that we had reduced adipogenesis in the 3T3L1 cells, very similar to the effect of LMPTP knockdown. So it appears that LMPTP promotes adipogenesis in vitro, and it's through an action through its catalytic activity. So we looked kind of over a time course of expression of adipogenic genes during this process in 3T3L1 cells. And what we noticed is that a very early transcription factor, CEBPB, we didn't see any effect on the expression over time. However, we looked at these later transcription factors that come up a little bit after CEBP and PPAR gamma and CEBPA, and we found substantially reduced transcription. Now, what's interesting is PPAR gamma promotes CEBPA expression, but it also promotes its own expression once it's expressed. So we thought, okay, perhaps this is the PPAR gamma is the point at which we start seeing an effect here. And we'll go more into that part later. We also looked at expression of some genes that are downstream PPAR gamma, such as adiponectin and fatty acid synthase, and you can see that there's a substantial reduction in transcription. So now we were left, since it was through the catalytic activity, we're left to what's the potential substrate or signaling pathway downstream of LMP2B. Now, LMP2B is known as being an insulin receptor phosphatase, and we had found that LMP2B dephosphorylates the insulin receptor in the liver during obesity, but we didn't see a difference in insulin signaling in the adipocytes or in the adipose tissue when we knocked out LMP2B. And also, when we're looking at the literature, we found that people have knocked out the insulin receptor in adipocytes, and that leads to lipodystrophy of white adipose tissue and insulin resistance, which is the opposite phenotype of what we'd expect if LMP2B was dephosphorylating the insulin receptor in the fat tissue. So we looked in more at some of the old LMP2B papers, and has been a group that worked a lot previously on LMP2B and fibroblasts, and they had shown that LMP2B negatively regulates the platelet-derived growth factor receptor, PDGF receptor activation in fibroblasts. And then there's also some literature showing that transgenic mice, which express a hyperactive mutants of the PDGF receptor, actually have impaired white adipose tissue development. And that if you treat PDGF or 3T301 cells in vitro with PDGF, that inhibits their in vitro adipogenesis. So we thought perhaps taking together LMP2B biology with what's known about the PDGF receptor in adipogenesis and adipose tissue, that maybe the PDGF receptor could be the substrate of LMP2B in this particular tissue. So we looked in 3T301 cells, and what we found is that the PDGF receptor alpha is actually very hyperphosphorylated in LMP2B inhibitor treated cells. And you can see, and we found this in resting conditions. So we looked at both PDGF receptor alpha and beta, and we did not see any difference in beta, but we saw a strong difference in alpha. And we found that we had an upregulation of phosphorylation of the PDGF receptor alpha in basal resting cells. So it appears that perhaps LMP2B could dephosphorylate the PDGF receptor alpha just in resting conditions. So we looked downstream to see if basal PDGF receptor signaling was affected. And so we looked at MAP kinases, and you can see we looked at PKC, we looked at some other pathways, AKT. The only thing that we saw an effect on were the MAP kinases, and we saw an increase or hyperactivation of both JNK and P38 in 3T301 cells subjected to the LMP2B inhibitor, but we did not see an effect on phosphorylation of ERK. So this was a little perplexing because we thought if we're having less adipogenesis, why would these MAP kinases be more active? And also why specifically JNK and P38, but not ERK? So we went back to the literature and actually found something interesting. So there were some pretty old papers that had shown that both the MAP kinases P38 and JNK regulate the activity of PPAR gamma by phosphorylating it on an inhibitory residue, serine 82. So actually activation of either P38 or JNK can lead to inhibition of PPAR gamma signaling. And given the fact that we saw a change in gene transcription at the PPAR gamma stage and later, but not with earlier transcription factors, we thought we should investigate this. So we looked at phosphorylation of PPAR gamma on this inhibitory serine 82. And as you can see, when you treat the cells with basal resting cells with the LMP2B inhibitor, you can see an significant increase in PPAR gamma phosphorylation on this inhibitory site, suggesting that PPAR gamma is less active. So then what we did was move into looking more at kind of the physiology of these cells. So we performed a metabolomics analysis with our collaborators, Stefano Tiziani, at the University of Texas, Austin. And so we treated 3T301 cells with the LMP2B inhibitor, and we collected the cells and their supernatants at various time points at days 2, 6, and 10 during the adipogenesis process. And at day 2, there's virtually none. At day 6, we're just getting started. And at day 10, we usually have full adipogenesis. And what we noticed is we did see some, in the PCA plot, some divergence between the DMSO and our inhibitor treated cells, which was starting at day 2, became more divergent at day 6, and spread out even more at day 10. And you can see from the volcano plot, we had a very nice distribution of differences, significantly different metabolites that were affected here. So we started looking into the different pathways that could be affected. And the most striking one that we noticed was glycolysis, because we noticed that there was, in the LMP2B inhibitor treated samples, there was a significant increase in glucose. So there's higher glucose, but there was a reduction in glycolytic intermediates, fructose 1,6-bisphosphate and phosphoenolpyruvate. But the final product of glycolysis, lactate, was significantly reduced, appearing that there was less glycolysis happening. Now, the first step of glycolysis is catalyzed by hexokinase 2 enzyme, which is actually, we found in the literature, was a PPAR gamma dependent gene. So we thought, well, if we have less expression of PPAR gamma or less activation of it, which leads to less expression, perhaps we have less glycolysis because of this. So we looked at the expression of hexokinase 2, and as you can see, we saw a significant reduction over time of the hexokinase 2 expression, which actually correlated well with the expression of PPAR gamma that we saw during the course. So it appears that we have a decrease in glycolysis, which we think is likely due to the reduction in PPAR gamma activation. So then we looked at, if there was less glucose, there must be an alternative energy source. So we looked at oxidative phosphorylation over the differentiation course. And what we noticed was a very, very striking difference in the ratio between the reduced to oxidized forms of electron carriers of the electron transport chains. And you can see this in NADH and both in coenzyme Q, that there was a huge reduction in the reduced form and a significant increase in the oxidized form, which suggests that there's actually more oxidative phosphorylation going on in the LMP2B inhibitor treated cells, which makes sense if they have a reduced glycolysis. Now, we had looked and found that the 3T301 cells are reported to have increased oxidative phosphorylation during proliferation. So, and then of course, during proliferation or other very energy intensive processes, cells upregulate the production of nucleotides. So, and we had found one of the first things that we had seen when we were analyzing the data was we had a huge increase in nucleotide synthesis, or nucleotides being present, which we didn't know how to explain at the beginning until we delved further into some of these pathways. And what we found is that pretty much across the board, we had a substantial increase in nucleotide levels and as you can see, of all the different nucleotide levels, which is suggestive of a high energy state because the nucleotides are needed to maintain that, which could be either from a proliferation or something else. But we think due to the effect on on the PDGF receptor that it's more of a proliferative state. So this is basically our model. Normally what happens in the 3T301 cells is, or any pre-adipocyte fibroblast actually, is that we think that the PDGF receptor alpha promotes the activation of MAP kinase pathways, particularly the P38 and J&K pathway, which would normally suppress the activation of PPAR gamma. So, and that would be anti-adipogenic. LMPTP, by dephosphorylating the PDGF receptor on an activation loop tyrosine, actually inhibits this process, which would eventually lead to more PPAR gamma activation and lead to mature adipocyte formation. And so we think that when we're treating with an LMPTP inhibitor, we're actually inhibiting this process. Now, during this process, there's a metabolic shift where we think that cells are going from a more proliferative state to an adipogenic state. So an adipogenic state is actually in contrast to a proliferative state. And so LMPTP helps promote that stop in proliferation and that promotion into a terminally differentiated state. Now, the conclusions for this project so far is that we found that LMPTP promotes adipogenesis, and it does this through its catalytic activity. And we propose that its substrate during this process is the PDGF receptor alpha. And so what we're proposing is a mechanism through which LMPTP dephosphorylates the PDGF receptor alpha, and that leads to a decrease in activation of MAP kinases and leads to but then reduced inhibitory phosphorylation of PPAR gamma, which is a key adipogenic transcription factor, both for transcribing genes that are important for the adipogenesis process, but also for transcribing genes that regulate cellular metabolism, which helps the cells shift from proliferating to going into a terminally differentiation. And our metabolics assessment suggests that LMPTP inhibition leads to metabolic shift during this differentiation process, which results in reduced glucose utilization and more oxidative phosphorylation. And so here are my acknowledgements. I've had the San Diegans from both La Jolla Institute, which is where I did my postdoc, and then UCSD, which is where I have my faculty position, collaborator, Dr. Nunzio Bottini, and my technician, Michael Diaz, and graduate student, Zachary Holmes, who did the bulk of this work that you see here, and also other members of my research team, Matthew Bliss and Vita Zhang. And then also our collaborators at the University of Texas, Austin, who did all of the metabolomics work that you can see here, Dr. Stefano Tiziani, his lab, and his graduate student, Megan Collins, and other members of his lab. And I would absolutely like to thank the 88 Pathway Award for basically giving my career start. I got this award as a postdoc. I got a faculty position at UCSD. I'm still involved in diabetes research and tremendously excited about it. And that wouldn't have happened without this award, and I'm extremely grateful. And I would also like to thank the UCSD UCLA Diabetes Research Center Pilot Award, because I got a pilot award specifically on this adipogenesis process. Okay, and with that, I'd be happy to take any questions or discussion.
Thank you so much. That was fascinating. Please, to my colleagues, ask any questions that you'd like in the chat, but I can go ahead and start. Mm-hmm. There's two things that really fascinated me here. One is this suppression of glycolysis, mm-hmm. which you think is at the level of hexokinase 2. Now, did you actually, sorry, it flew by a little bit. Did you actually demonstrate that hexokinase 2 is downregulated and that glucose 6-phosphate has been suppressed among the metabolites that you measured?
Right. Okay, that's a great question. So first of all, we did show that hexokinase 2 expression is reduced. Now, and then the other question you had was with the glucose 1,6-phosphate?
No, glucose 6-phosphate.
Glucose 6-phosphate, yes. So we actually—
I didn't see it there. That's why I'm asking.
We did not detect that in the metabolomics analyses. However, what we had found that I didn't show here was that we found reduced intermediates during the pentose phosphate pathway as well. And since glucose 6-phosphate is actually the first step at both of those pathways, that's where, and we got to the hexokinase 2 because it looked like both glycolysis and the pentose phosphate pathways were down.
Well, then that actually short circuits my real question, which was the fascinating change in the broad array of nucleotides. And it's not just the triphosphate nucleotides, it's the monophosphate and diphosphate.
Right.
And I actually was wondering whether what you had actually done is divert carbon into the pentose shunt, because as you know, ribose 5-phosphate is part of the building blocks for nucleotides.
Yeah.
And would that have explained this across the board change in nucleotide pools? But maybe not if you really have a clear down regulation of hexokinase 2. Yeah, but I'm still puzzled by this observation. If glucose utilization is so sharply curtailed, specifically by hexokinase knockdown.
Right, right. The nucleotides was difference was the biggest surprise to us as well. So, and we're still working on this and don't have a full explanation or model of how metabolically the nucleotide synthesis is upregulated, because it's clearly across the board in every type of nucleotide, which
Right.
is which was, I'd say, our most striking result.
And then you'll forgive me this question. I don't think you said what is the fuel source for these cells if they're choked off from glucose. I don't think you actually came out and said what is their source of metabolic substrate. And have you thought about branched chain amino acids? So sorry to be a homer here, but there is excellent literature about differentiation of 3T3 cells involving maturation of branched chain amino acid metabolic pathways.
It's funny because we had been wondering that too, and after seeing Phil White's talk, it made me think that actually that's something that we should look into branched chain amino acids for this particular phosphatase.
Yeah. Okay. All right. Any other questions from folks? I thought that was really clear, really elegant presentation, and a lot of really interesting results. Congratulations on that. And just quickly, our next session is on August 4th. Philip White and Judith Agudo will present that day. I'm assured from both of them that they'll have a lot more to say than they did in their recent ADA talks. So I don't think we should worry about rapid reappearance and repetition. There'll be plenty of new things. So please join us then. And perhaps more importantly, I apologize for anyone from England on the call. Go Denmark right now, starting the Euro contest. So thanks everybody. See you on August 4th, and thank you to our speakers. Terrific job. Thanks very much.
Thank you, and thanks for organizing.
Okay, cheers. Bye-bye.